All right, well, thank you so much for coming out here today. It's a thrill to be here with you. So, as you just heard, I'm a plant biologist. I study the evolution of genetic programs that control the development, particularly of flowers. Now, I have to admit, I've sat next to a lot of people on planes and told them that I was a plant biologist. And often their reaction is, you know, help me find out why my fern is dying or something like that. I don't keep things alive. This is the problem. I like to grind them up in little test tubes and things like that. So don't ask me how to keep something alive. But I find that we talk in plant biology about plant blindness. People often don't appreciate the importance of plants in our everyday life. But I think we can all agree that flowers are very compelling and charismatic. As a human civilization, we've had a fascination with flowers really from the beginning. And what we're really trying to understand in our lab is how we have, over the course of evolution, generated this enormous diversity of floral morphology, and particularly how changes in the genetic programs that control these developmental processes has generated the change in morphology. And in order to do that, we work at the intersection of sort of three different fields. We ask different types of questions. So, for instance, we're very interested in development, very specifically. What are the patterns of cell division and cell expansion, the duration, their localization, and how do these different patterns generate the morphology that we're interested in? So we ask very specific questions about how morphology changes over time. And then we overlay this information with information about the genes. How do different genes interact with each other on a local scale, on a global scale, on the genomic scale, and ask how these genetic programs control the developmental processes. And then we take all of those questions and we put them in an evolutionary context and say, well, when we ask all these questions in one species and we compare them to a different species, what aspects of these processes are the same, what aspects are different, and can we understand how those differences arise? So when we put all those things together, that's a field called evo-devo, or evolutionary developmental biology. So we're essentially asking how morphological diversity is generated over time at a molecular level. So why do we use plants as a model system? So if you'll let me proselytize for a moment about why plants are such a wonderful model system. I'm a developmental biologist. If I was an animal developmental biologist, I would only be focused on a very narrow window of that organism's life, what we typically call embryogenesis, from fertilization to some type of hatching. But in plants, the fantastic thing about plants is that if they are alive, they are undergoing development. If we look at this plant embryo, you can immediately recognize that while, yes, there was a developmental process that took this from a single cell to this embryo, this is not the entirety of the body of a plant. So this enormous oak tree started out as this tiny embryo, and it is in a constant state of organogenesis and producing new organs in order to generate its body. And all around us, I mean, we finally got to go outside today and enjoy a little bit of nice weather. You can see trees all over campus, things coming up from the ground. These are plants that are undergoing organogenesis even in what we would consider their adult body. And plants can do that because all throughout their body, they have reservoirs of undifferentiated cells, stem cells. Now, my animal colleagues get really excited about stem cells. Stem cells are nothing for plants. They've got stem cells to spare. Their stem cells are arranged in these specific regions called meristems. They're domes of undifferentiated cells. So when I tell you that I want to understand floral development, this is where we start. We start with a dome of undifferentiated cells, and we look at how that set of cells changes over time to generate floral morphology. So this is our big problem. We have about 350,000 species of flowering plants that have arisen over the past 150 million years. That's a big problem. I'm not looking at that scale, right? You have to start with something tractable. And what we work with is a model system called Aquaregia that you may know as columbine. A lot of you may have this in your yards at home. It is what we call a recently radiated species flock. So let me explain to you what that is. Why is Aquaregia a good model system? So obviously it's very pretty, but it's also very tractable from a molecular standpoint. It has a relatively small genome, about 300 million base pairs, and you'll take my word for it, that's manageable. We have a fully sequenced genome. We also have a lot of functional tools that allow us to trick the plant into doing things that we want it to do. It also has a wonderful array of morphological novelty. So I mentioned that this is a radiated species flock. What I'm talking about? I'm talking about that when we study the evolutionary history of this genus, when we reconstruct its phylogeny and understand how the species are related to each other, we see that these species have radiated over a period of 1 to 6 million years, which is relatively recent. And in fact, the North American radiation that's pictured here is less than 1 million years. So these are very closely related species. That means that they're also interfertile. And that's something that makes the geneticists sit up and take notice, because we can cross these species, even species that look quite different from one another. We can hybridize them and then look at how their traits segregate in later generations and map the molecular changes that are responsible for the differences in morphology. So this means that we have traditional developmental genetic tools, and we also have a lot of evolutionary genetic tools that make it a good model system. Okay, so let's step back a second. I'm going to tell you a little bit about floral morphology and floral development. So this is what you usually think of when you think about a flower. You may have had a little bit of plant biology, maybe in elementary school, and they told you that there are 4 different kinds of floral organs, right? So you have sepals that are the productive organs, the attractive petals that are involved in manipulating pollinators, the male stamens, and the female carpels. And these organs are arranged in what we call whorls. They're concentric circles. So moving, you get sepals, petals, stamens, and carpels. And about 30 years ago, molecular biologists working in a number of different model systems defined a genetic program called the ABC model. And this is a model that tells us how each of these floral organs figures out during development what flavor of organ it wants to be. So for instance, there are 3 different classes of genes, the A, B, and C class genes. If you're primordium in the first whorl, you're only exposed to A function, and that tells you to be a sepal. In the second whorl, you have A and B function, that tells you to be a petal. Stamens are exposed to B and C together, and then carpels to C alone. So this is what we call a combinatorial code. Each little position in the floral meristem has a different set of genes, and that tells the organ what kind of organ to be. And the way that they came up with this program was by examining mutants. In developmental geneticists, we love mutants. We make mutants all the time. We like to knock out gene function and then infer from the way the plant looks, or any other organism, the way it looks, what the function of that gene must be. And the genes that comprise the ABC program have something in common, that when you knock them out with mutation, you dramatically change floral morphology. You transform the identity of one organ into the identity of another. And so here, for instance, if you eliminate this B class gene, then all you have are A and C functions in the floral meristem, and the petals turn into sepals, and the stamens turn into carpels. And this is a complete transformation. So this highlights how minor genetic changes can have really profound effects on morphology. So these are the kinds of genetic programs that we think about when we think about floral evolution. Okay, so how are we applying this kind of model in the context of our model system, Aquaregia? So there's a lot of interesting things about Aquaregia flowers. These guys up here, those are the sepals. And usually you think about sepals as being green and protective, but these are not just green and protective. They're bright red, or in some cases blue or white or yellow. They're what we call petaloid, which just means that they're showy. So this is an interesting question. Here we have the ecological function of pollinator attraction that has been transferred from the inner whorl of the flower to the outer whorl of the flower. So how does that happen, and what's going on at the molecular level in order to make these sepals bright and showy? Another really interesting aspect of the Aquaregia flower is this nectar spur. So the true petals in the second whorl have this long tubular structure that terminates in a nectary. And you can just imagine all the things that Aquaregia is doing to manipulate pollinators to make them go all the way down here and get the nectar. So there's all kinds of things that we can study here related to how the spurs first evolved. They're what we call a key innovation that were important for the radiation of the genus. And what is the genetic basis of their diversification in morphology? They get longer and shorter, and they're curved, and they have different shapes. So we're studying all these different aspects across species of Aquaregia. And then one of my favorite things about the Aquaregia flower that I didn't even realize until I started dissecting them is that they actually have 5 types of floral organs. They have this 5th type of organ called the staminodium, which is a sterile organ positioned between the male stamens and the female carpels. So how the heck does that work? The ABC model gives us a very elegant mechanism for giving us 4 types of floral organs. What happens when we intercalate a 5th type of floral organ into that genetic program? So we've been studying all of these questions. Another one that I'm going to show you today is we've been interested in this question about how does the flower know when to stop? You think about a flower, once it makes those carpels, then it stops. It doesn't make any other floral organs. And that's actually a very carefully controlled genetic process. In most of the flowers you might think of, there may only be 1 whorl of stamens, like in this snapdragon, but here we're looking down on an Aquaregia flower, you can see that there are many whorls of stamens. So how does the meristem decide how many whorls it's going to make? And to investigate this, we started by actually working with these C-class genes, because, and this makes sense when you think about it, it turns out the C-class genes confer the identity of the stamens and the carpels, but in conferring identity of the carpels, they tell the floral meristem to stop. So what we want to ask is what happens when we knock out the function of these genes? And in Aquaregia, we use a molecular tool to trick the plant into silencing our gene of interest. And when we do that, here's a wild-type Aquaregia flower, and here's a flower where we have used this trick to silence the C-class gene. And you can already see it's looking a little weird, but as it develops, you see that the stamens are completely replaced by petals, and the staminodia, that novel organ, and the carpels are both replaced by sepals. And when we look inside the flower, you may not be able to see this, but there are actually dozens and dozens of whorls of organs here. So it's clear that this gene also controls floral meristem termination. Now this was actually just the first step in this project. We were trying to confirm that this gene functions the same way in Aquaregia that it functions in other species. We expected it to be conserved because in fact all around you see mutants. These are all mutant varieties. So here's a rose, a daffodil, and a peony, where on the left you see the wild-type flower that has a single whorl of petals and many whorls of stamens, and on the right you see an agamous mutant that has the transformation of stamen identity into petal identity and the indeterminacy of the floral meristem. So actually you're looking at mutants all the time, you may just not have known it. So homeotic or transformative mutants have always been very popular in horticulture. So I hope I've convinced you that Evo-Devo is an interesting field. If you're not totally taken by plant biology as I am, you might be interested in some of my other colleagues, Mansi Srivastava, who studies these fascinating little flatworms that have the capacity to regenerate their entire bodies from just small fragments of their body. They're actually thought to be immortal. My colleague Cassandra Extravor, who studies the evolution of the genetic pathways that control germline specification, which is incredibly important for evolution and development. Cliff Tabin over at the medical school studies the diversification of vertebrates, and particularly the vertebral column itself. Why don't snakes have any limbs? Why do ostriches have such long necks? My colleague Terry Cappellini over in human evolutionary biology is actually using Evo-Devo approaches to understand the evolution of the skeleton in humans. So he uses mouse models a lot, but what he's ultimately interested in is actually human skeletal evolution. So that's everything I'm going to tell you today about Evo-Devo.